My name is Crystal Dilworth. I'm a neuroscientist with a PhD from Caltech in Pasadena, California. These are unprecedented times. The outbreak of the novel coronavirus has the world in its grip. Not since the Spanish flu of 1918 has a disease spread across the globe so far and so fast.
Today, scientists have a high-tech toolkit to attack. From viral tests to the study of antibodies to vaccines, we're all dealing with the new vocabulary and complex problems.
But how did we get here? And what have we learned? This is the story behind the virus called SARS-CoV-2 that has left a trail of infection touching everyone, everywhere.
Powering the future. Confronting the world's challenges with cutting-edge solutions, medical breakthroughs, and high-tech discoveries. These are the stories of our planet. This is VOA Tech.
I'm going to swab your nose for the coronavirus. Would you please have a seat? Now, this shouldn't hurt. It's just a little tickle.
Okay.
All right. I'm going to do the other side.
It does tickle. Los Angeles, California. This is a demonstration of the much coveted coronavirus test. A swab of the nose, a sample now taken by medical professionals in full protective medical gear. This was filmed before the pandemic outbreak would take hold. This is when hospitals, like this one run by the University of Southern California, were merely getting prepared. They would soon be in a full-on fight.
All the patients in this room, all the feet that you see, they all have COVID.
This video, produced by the New York Times, chronicled the disease surge in New York, the city that became the COVID-19 epicenter in the United States.
So you're the one that gets the call.
I am the one who gets the call. I am here 24/7 available.
In Los Angeles, Patricia Sung oversees infection prevention at USC's Verdugo Hills Hospital.
When a patient comes in, how do you test to see if they have coronavirus?
We have supplies already in stock that we use for other respiratory virus panels. The only difference is the type of test that's performed on it. We basically follow our local public health strategy for highly communicable diseases like measles, like TB, like chickenpox that we encounter very regularly in our community. Our nurses would collect a swab. This is a viral respiratory swab. They would collect one from the nose, the nares, and a separate one from the mouth, from the throat. And those would be sent to our public health lab for testing for the novel coronavirus specifically.
Thank you. I'm going to send this to the lab now.
So it's the same procedure for any respiratory illness. It's just the test that's performed afterwards that's different?
Yes, that is the only difference.
COVID-19 may be handled with the same safety protocols, but there's a big difference: volume.
We hear that there's not enough testing kits. What does that mean?
So that means that the chemicals that are used to detect the presence of the virus in the swabs that we're collecting can sometimes not be enough or not have the right amounts. And so the CDC is working to ensure that all the test kits that come out are able to properly identify if a patient has the novel coronavirus or not.
So it's not necessarily swabs and kits like this, but it's the actual reagents that are used to process the samples.
Correct. It's the ability of a lab to detect the presence of the virus as opposed to the collection of a respiratory specimen that would be used to detect.
The medical system in Los Angeles and across the United States has little to compare to this event. Sung showed us what she had in place in early March 2020, and this emergency room is considered well-equipped. Still, there is only one room where a suspected infected patient can be isolated from others.
So a patient under evaluation would be brought into this room, placed on the bed, and connected to our monitor, similar to patients under evaluation for any other respiratory illness. The difference would be that we would turn on our negative pressure filter here that basically sucks up all the air and sends it to the outside before it gets irradiated with UV light to disinfect the air, and it would protect our patients outside.
So can we turn it on?
Yeah, we can turn it on.
Okay.
So our staff would bring our patient in, and prior to coming in, they would just turn this knob, You hear a low whirring noise. That's the filter working to suck all the air up into the vent.
So you've got air going through a filter, it's being cured by UV, and then it's being jettisoned out of the building.
Out of the building.
Okay, so get all of those germs away from us.
Get all the germs out of the room so we don't have exposure to our staff or to our patients, and we kind of help keep our patients safe as well.
The idea in this ER and others around the country is to isolate suspected COVID-19 patients. Mary Virgilito is Director of Patient Safety here.
So if the staff identifies that the patient has some of these risks, what happens next?
So immediately the patient is handed a mask. A nurse is actually right around the corner, the triage nurse. They will pull the patient in through and actually walk them down this hall. And then we would enter the emergency department right here. It's a straight hallway down to our isolation room, our airborne isolation room, where the patient would be placed, made comfortable, and then assessed for whether or not they really are at risk for coronavirus and if we need to do testing and contact.
Much of the ER's first line of defense is made up of infection-fighting bleach wipes, sanitizer, and hydrogen peroxide. The ER teams know these old standbys are their best protection. They would also need better protective gear as the pandemic took hold. Shortages hit American hospitals and frontline healthcare workers hard.
So would the medical staff that are treating a patient in a room like this be wearing anything different, doing anything different to protect themselves?
CDC has recommendations for healthcare workers to protect themselves against possible coronavirus patients. And so they would be wearing a gown and gloves to protect from the contact with the patient. They would also be wearing special masks. These are respirators that help filter the air before they breathe. And they would also be wearing eye protection to make sure that it protects them against any possible secretions or spray from the patient as they're providing the care that they need.
So I'd like to introduce you to Luna. Luna is our UV disinfection robot.
How cool.
Yeah, and Luna is actually part of our plan for a coronavirus patient. So after a patient was in the room, we want to disinfect it and get it ready for the next patient.
Hospitals also use technology.
So you said that she spreads UV. How does she do that?
So it's kind of like an R2-D2 setup. When she's in the room and there's no one there, this top pops up. And actually there's all of these lamps that spray out 360 degrees of UV light. And it flashes kind of like a disco. UV frequency, this is UVC. And UVC is actually blocked by our atmosphere. So viruses and bacteria have no defense against it because they're not used to seeing it.
The fight against the coronavirus is now global. What began with a few cases of an unusual form of pneumonia quickly spread. Wuhan, China, December of 2019. Chinese authorities informed the World Health Organization of a mystery illness. A week later, Chinese scientists confirmed the cases are a novel coronavirus. January 10, 2020, they released the virus's DNA sequence to scientists around the world. It's too late. Asia is hit first. South Korea mounts a massive disinfection and testing effort. But travelers from China are already spreading the virus. Europe and the United States report cases. The Mideast would be next. Iraq tries to stop the virus at border checkpoints. Iran is hit hard. In early March, Italy would be locked down. All the major tourist attractions close as the death toll mounts. March 5th in the US, a dramatic scene. The California Air National Guard drops coronavirus test kits down to a cruise ship quarantined off the coast of San Francisco because the disease is believed to have spread on board. The epidemic is growing. March 11, 2020, the World Health Organization makes a stunning declaration.
We have therefore made the assessment that COVID-19 can be characterized as a pandemic. Pandemic is not a word to use lightly or carelessly. It's a word that, if misused, can cause unreasonable fear.
This particular coronavirus that's causing what we are calling COVID-19 is actually technically called the SARS-CoV-2 virus.
SARS-2 sounds like a sequel I don't want to see.
Yeah, that's right.
Robert Kim Farley is an epidemiologist who studies the spread of infectious disease for the University of California, Los Angeles.
So the world is concerned about coronavirus. But first, I'd like to know, what is a virus? What is a coronavirus? Why are we worried?
Yeah, so basically the coronavirus is indeed a virus. It's not a bacteria, it's a viral particle. It can be spread from person to person. And why we're concerned is that this coronavirus is among the family that also had the severe acute respiratory syndrome virus, SARS, or the Middle East Respiratory Syndrome virus, MERS.
SARS, Severe Acute Respiratory Syndrome, and MERS, Middle East Respiratory Syndrome, are both pathogens from the coronavirus family. SARS left a deadly path marching across Asia, North and South America, and Europe in 2003. MERS spread mostly through the Middle East in 2012. They were deadly, but neither was a pandemic like COVID-19.
So we were talking about transmission of the disease, so vocabulary words that we hear: outbreak, pandemic, epidemic. Help me understand.
Basically, the word outbreak is when you have more cases of a disease than you were expecting to have. So that's what we call an outbreak. An epidemic is then when you have a lot more cases in a lot larger area, oftentimes affected. And then the word pandemic is reserved for when multiple continents and multiple countries are being infected and have what we call local transmission going on. I kind of put it to the concept of a matchbook with matches, okay? And you're just striking matches and you're throwing them into the woods. Some places that's going to catch fire.
In the lab, scientists were able to quickly isolate the novel coronavirus, SARS-CoV-2, and map its structure. These images were taken by extremely powerful electron microscopes. It allows scientists to study and understand the virus. Early on, they learned an important fact about its genetic source. The director of the U.S. National Institute of Allergy and Infectious Diseases, Dr. Anthony Fauci, explained how COVID-19 spread
during one of the many coronavirus task force briefings held during the crisis.
So these are the kind of things that go from an animal reservoir to a human. Sometimes it doesn't go anywhere, it just goes one or two people. But sometimes it adapts itself to function very, very, very efficiently.
How did it start?
So in terms of the epidemiologists looking back and seeing where these cases first originated in Wuhan City, actually about 2/3 of the first 41 cases that were discovered came and had association with a what they call a wet market, where live animals are sold, and oftentimes exotic animals. And so that may have been the epicenter, if you will, of where we started. It appears that that coronavirus is actually has a lot of bat coronavirus characteristics. So it may very well have come from a bat.
So when you're looking at the structure of the viral RNA, you see bat sequences?
Correct, exactly right.
These are the labs of the prestigious La Jolla Institute for Immunology, just outside San Diego, California. They're part of the front line of research going into finding a cure for COVID-19. Postdoctoral researcher Mike Norris is working on replicating the virus.
Okay, so what we've done here is these cells are expressing the protein that we're interested in. So when we spin them down, and what you can see is there's two layers here between them. The darker green layer are the ones that are expressing the protein that we want. And so what we do is we then dump off this supernatant layer, and then we plunge freeze the cell pellets into liquid nitrogen, which will freeze them very rapidly and allow us to then purify them further later. So freezing this actually slightly disrupts the cell integrity, which actually allows us to get the protein out of them much easier. So we snap freeze them in liquid nitrogen, which rapidly freezes them. And then when we thaw them, they'll start to break up. And then we use another methodology to then break up the actual cell and release the protein that we want.
We have to expect that these kinds of pandemics are going to come again and again and again.
Erica Ullman-Sapphire is a professor at the La Jolla Institute with a background in structural biology and immunology.
So what I was working on today is a structural protein of the virus. It's actually the puzzle pieces that build to make we call it the viral particle, that actually make the virus itself. By working on other viruses, we've developed an expertise in learning how to handle that kind of protein. and how it interacts with your human membrane to build the virus. And so today what we have been doing is making copies of that molecule in order to see how they build.
So how does viral infection actually work?
What happens is that the surface molecule in the virus, and it's shaped like a knob, kind of like a mushroom on a surface, that looks for a certain human molecule that is its receptor, like a key for a door. And when it finds that receptor, it will attach. And after attaching, there's a mechanism like a spear fishing rod, whereby the virus springs forward something that'll drive itself into the human cell and fuse its way in. The good news is we understand those kinds of processes, and we can develop molecules to stop them.
Ullman Sapphire has fought extremely dangerous viruses before, like Ebola, the devastating outbreak that struck West Africa from 2014 to 2016. More than 20,000 people got sick. 11,000 others died. Ullman Sapphire was part of an international consortium of scientists who came up with a way to stop Ebola using antibodies. She hopes the same concept can work with the novel coronavirus.
One of the things we're doing here is we're leading an international effort to develop antibodies against this virus and compare them. So an antibody is a molecule made by our own human immune system. So if you've been infected with something and cleared it, you have antibodies against that virus that can now find it and attack it. If you've had a vaccine that gives you protective immunity, that vaccine gave you antibodies. So antibodies are molecules from your immune system that are very precise. They have like fingerprint recognition for a certain place on a certain pathogen. If you can find the right antibody, you can use it as a drug to directly inactivate that pathogen.
So if we can identify the right antibody against COVID-19, we can start to prevent future infections before a formal vaccine is developed.
Exactly. By finding the right antibodies now, we have a drug that we can use to treat people that have gotten sick. We have something we can use to protect those frontline medical workers.
Back in the lab, the culture is ready for the next step in the process. Liquids are separated. Once they're precisely balanced, they're ready for the centrifuge.
We're asking those cells that we're growing in culture to make copies of this virus protein. It's very safe because they're just copies of one protein by itself. We're going to break these cells open to release that molecule, and then we're going to purify it using different columns and different strategies where it flows over a matrix in order to separate out impurities and get just the molecule we want, one that's highly purified. And then if we can take those powerful high-resolution photographs of the molecule in every possible orientation, then we can computationally reconstruct the three-dimensional form. And we can even get to the detail of seeing how that polypeptide is knit together in space, sort of how that piece of yarn makes that sweater, and what's a sleeve and what's a neck, and how the thing works and where the buttonholes are.
Unlocking that key is what this lab and many others here and around the world are working on right now, testing antibody resistance to COVID-19.
There are different ways to make antibodies. One way is to look at the blood of people that have survived, because their own immune system has evolved those molecules, and sometimes there's something extraordinary in that person's blood that could be a drug to help others. So we've done that a lot, and that's a great way of doing it. If you don't have many survivors, or you don't have many survivors yet, you can do different things. You can make chemical libraries that express billions of possible human antibodies in little test tubes. And you can survey those and make little changes and chemically find the thing you want. You can also immunize special mice that can make the right kinds of antibodies. And so there are lots of different strategies, and there are lots of organizations using every one of those right now.
Seems like direct from patient would be the most straightforward.
Direct from patient is most straightforward, and you can often get the extraordinary molecules that way. The issue is time, that you need the patient to have already survived, and it might take a couple of months after they've survived for their immune system to keep evolving and creating that best antibody response. And so that's great if you have a couple of months. But you don't have a couple of months, we're going to use some of these other earlier strategies, and I think the right thing to do is pursue them both at the same time.
LJI is also part of a group of laboratories around the world looking at possible vaccines. Shane Crotty is a professor here. He specializes in human response to vaccines.
Essentially, there are 28 vaccines currently, and there are probably thousands of viruses and equivalent numbers of bacteria, and then there are the parasites on top of that. So there are many more pathogens than there are vaccines. So of those 28 licensed vaccines, most of them took 20 years plus to actually understand the problem well enough and then actually get something made that was in people.
I think we're hoping for a little bit less than 20 years.
We're hoping for faster, right? And so I started my lab 16 years ago with that in mind, that, okay, vaccines are incredibly valuable, but it's a very slow and uncertain path. Can we really do better to understand fundamentals and turn it into a more engineering and reproducible thing?
As of April 2020, there isn't a vaccine for the novel coronavirus.
SARS-2 coronavirus, COVID-19 disease, is a good candidate for the type of disease for which one could optimistically think a vaccine path would be relatively fast, and that's because it's an acute infection that most people, their immune system handles it fine. They recover from the infection relatively quickly. So in general, diseases like that are diseases that's an indication that your immune system is making a good antibody response, is making a good T-cell response. And so a vaccine that mimics that would have a good chance of generating a protective antibody response or a protective T-cell response that would get the job done.
March 16, 2020, Seattle, Washington. The first human testing begins as a volunteer is injected with a coronavirus vaccine candidate. A second simultaneous study at Emory University in Atlanta is started March 27. The vaccine, mRNA-1273, was developed by Massachusetts biotech company Moderna. It contains a short segment of RNA from the virus. This first vaccine to be tested will most likely not be the last. Dr. Crotty explains what scientists are looking for.
I'm trying to get what we might call sort of good inflammation and not bad inflammation. How do we get a high antibody response instead of a low antibody response? And how do we get memory? How do you get the immune system to remember what you just immunized it with? There's a big toolbox, both an immunological toolbox, a vaccine components toolbox, and infectious disease or sort of pathogen analysis toolbox.
Whether it's an antibody therapeutic treatment or development of a vaccine, the research community has never been more focused on a single target. One thing we know, this simply isn't going away.
We have to expect that these kinds of pandemics are going to come again and again and again. We travel more than we ever did at any other point in human history. We are encroaching in wild areas. As the climate changes and insects move, we can expect to see more and more infectious disease. It is, in my opinion, easier and faster to mobilize a vaccine or a drug and to solve this problem than to tackle some of the more complex issues that are the reasons why these viruses are emerging and reemerging. It's hard to change human behavior. We will continue to travel. We will continue to encroach on wild areas. We will continue to interact with each other. We will continue to live in cities. It's just going to happen.